Today I'm going to walk through some of the considerations and best practices for panel design for the iQue HTS instrument. This is one of the most critical elements for designing a flow cytometry experiment, particularly when using large complex mixtures of antibodies. Proper design will ensure that the panel of antibodies and fluorophores used will generate the best possible data from your sample. In this example from the violet, blue, red or VBR iQue instrument, you can see that there are 3 lasers which excite different fluorophores depending on their excitation wavelength. The iQue HTS has 13 detection channels for fluorescence, which can detect different fluorophores based on their emission spectra. The bandwidth for most emission filters is shared for detection of signals following excitation from multiple lasers. For example, the 635 or 30 nanometer filter is shared across VL5, BL1 and RL1 channels. There are a huge number of common fluorophores and dyes that can be detected across the range of iQue instruments, with various choices per laser and detection channel. There is a useful fluorescent spectral viewer that can be found on the Sartorius website that can be used to determine which iQue channel will be the best fit for a particular fluorophore. You can select which instrument you have from the drop-down list, and this will bring up the wavelengths of the excitation lasers. You can then click Normalize onto each excitation laser to bring up a visualization of each detection filter available from that laser. In this example, I have selected the FitC fluorophore from the list and can see from the dotted line for the excitation spectra that it is strongly excited at the 488 nanometer wavelength of the blue laser. I can also see that the shaded region for the emission spectra crosses nicely with the 530-30 nanometer detection filter, also known as BL1 in the VBR iQue instrument. This shows that BL1 is the optimal channel to detect this fluorophore in. If we add in a second fluorophore, for example PE, which fits in the BL2 channel, we can get an idea of what the level of overlap is between the emission spectra of the 2 fluorophores. Ideally, we want to minimise the overlap between the emission spectra of fluorophores. Therefore, if only using a few fluorophores in a panel, it is best to spread them out across the lasers and detection channels to reduce the likelihood of overlap. If designing a larger panel, it becomes more important to select the optimal fluorophores and detection channels for each of your markers. One important thing to consider is the strength of detection in each of the iQue channels. Generally, the shorter wavelength channels such as VL1, BL1 and RL1 will produce the strongest signals, while the longer wavelength channels such as VL6, BL5 and RL2 will result in weaker signals. It can therefore be advantageous to use the stronger detection channels for detection of rarer, lowly expressed markers or for core markers that are most critical for the analysis. Another important consideration is the brightness of the fluorophore used. Fluorophores can be ranked based on their stain index, which is the separation ratio between the positive and negative populations. This parameter may vary based on the type of cytometer used, the antibody clone, and the buffer conditions. This example was generated by assessing the brightness of various dyes conjugated to a CD4 antibody and analysed using the iQue HTS platform. Generally, it is preferable to use brighter dyes to quantify lowly expressed markers and dimmer dyes for high expressed markers. For example, CD3, 4 and 8 are typically highly expressed and so could be stained using a dimmer fluorophore. Conversely, CCR7 is a weakly expressed marker, so may benefit from staining with a bright fluorophore with detection in one of the stronger channels like VL1, BL1 or RL1. To compare stain index more accurately for your cytometer, and to determine the optimal concentration of antibody for your experiment, a titration can be performed to determine the conditions with the highest stain index, and therefore the lowest background for your antibody. The final consideration that we will mention today is that if you are staining immune cells, we want to ensure that the antibodies are only binding to cells via their target receptors, and not via the Fc receptors on the immune cells. To avoid this, an Fc blocking solution can be added to the cells during the staining procedure. Even following the best practices for panel design, if you're using a complex multicolour staining panel, there is still likely to be some overlap between the emission spectra of the fluorophores, which is addressed using compensation. We have created a separate video to walk through how to run compensation of the iQue HTS platform, for which a link is provided.